Hey everyone, apologies for the long break between the last video and this one. Life kind of got in the way. But I'm bringing you another video today about a solution to Peto's paradox. And this time it's not about elephants, it's about whales, specifically bowhead whales. And I hope this is another really illuminating example of how we can learn a lot about cancer biology by comparative physiology between species. So let's get into it. So the video today is about a species called bowhead whales. And bowhead whales are fascinating because not only are they some of the largest animals that have ever swam, the Earth's oceans, they're also some of the longest lived animals. And they used to be hunted quite prodigiously by humans because of their propensity to have a lot of blubber on them. And blubber was actually a major source of fuel and oil, prior to the discovery of fossil fuels like kerosene. And so, bowhead whales were actually almost hunted to extinction in the early 19th century. Here's a painting, for example, of a bowhead whale hunt. So a very grueling endeavor. And so how do we even know that bowhead whales can live this long? I mean, who's been tracking that for this long? Well, it's actually a fascinating story. And that is that there were certain bomb lances used in the 19th century to hunt these unfortunate whales. And some of them actually survived encounters like this. And so we find that sometimes bowhead whales, have embedded lances from like the 19th century, 200 years ago, embedded deep in their flesh, which means they were shot, and then their flesh and tissue actually grew around that wound. And so that's how we know that some of these lances are so old, have been radiocarbon dated, that we can realize that bowhead whales can live for a fascinating amount of time. And it's very fascinating because they also survive these encounters. So this is an interesting way. And there's other techniques that show us that they can even live longer than 2 centuries. So very fascinating species in general. And so then this establishes two dilemmas for us for examining Peto's paradox within bowhead whales. Because not only are they some of the largest animals on Earth, weighing up to 60 tons, which is just insane, they can also live for such a long time, double the lifespan of an average human, the double lifespan of, some very geriatric humans as well. So not only do they have a lot of cells in which to get oncogenic mutations, but they have a lot of time in which they should get oncogenic mutations and develop some really fulminant cancers at that point too. And yet, controlling for both of these factors, they have a very low observed cancer incidence rate. So this really begs the question, what are they exactly doing right? Well, the paper that I'm discussing in this video today came out in late October 2025, and it's titled Evidence for Improved DNA Repair in the Long-Lived Bowhead Whale. And you can see that was published in Nature, so a very reputable source. And interestingly, in comparison to the last video I made about the solution to Peto's paradox in African and Asian elephants, this is actually a completely different strategy in bowhead whales as compared to what we see in elephants. Because while elephant cells are generally more sensitive to programmed cell death or apoptosis, because again, they express multiple copies of these genes like LAF6 and TP53, the fibroblasts, or the fiber-containing cells of bowhead whales, are not similarly prone to apoptosis following genotoxic stress. And they have low basal rates of TP53, so that begs the question, how exactly are they preventing cancer? And actually, just as another addendum, they actually require fewer tumor suppressor hits for oncogenic mutation. So they're actually more predisposed to oncogenic mutation than we'd expect normally. So again, this is building up into another kind of multiple layers of this paradox in whales. So number one, they found that bowhead whales have, in vitro, have less single nucleotide variants, small indels, and large structural variants. So they have less of these kind of genetic architectural changes as we'd expect, which is interesting because they do have a higher propensity to acquire an oncogenic mutation. And number two, they have enhanced double strand and mismatch repair. So if you remember, double strand repair and mismatch repair are really powerful techniques for how the double strand genome and all eukaryotic cells are usually repaired. Because of course there's a lot of mismatching that goes on, there are structural imbalances. It's very important to kind of repair these. Mismatch repair is particularly common when you think about, for example, the oxidation of certain nucleotides, the deamination of certain nucleotides that creates, constant switching between, for example, thymidine and uracil and cytosine, for example. You also see this with adenosine and guanine too, so it's very common in general. And the reason in bowhead whales is because they have something called enhanced poly-ADP ribose polymerase activation, and they have that after you introduce genotoxic stress via hydrogen peroxide and gamma radiation. And these whale fibroblasts, they have higher frequencies of non-homologous end joining and homologous repair in response to introducing some chemotherapies that are supposed to increase DNA mismatch, such as bleomycin and etoposide treatment. So this is also very interesting. And then finally, whale fibroblasts have a more accurate non-homologous end joining. And remember, non-homologous end joining is a way to repair double-stranded kind of discrepancies. And that can be inaccurate, but they have more accurate NHEJ. And they have, for example, after CRISPR, which naturally introduced double-stranded breaks, they have a higher fraction of unmodified alleles, which means their NHEJ is very accurate and very sensitive to detecting these inaccuracies and fixing them. So what contributes to these three features in whale cells? Well, the key kind of character here is something called cold-inducible RNA binding protein, or CIRBP. This is a stress-responsive RNA and poly-ADP ribose binding protein, similar to PARP. And again, PARP is actually used, or it's actually targeted within the context of some cancers, like myelomas in humans, and that's to kind of suppress the natural DNA corrective ability of these cancers to accelerate their death. And the human and whale CIRBP protein only differ by about 5 amino acid residues, but what a difference those 5 residues make. Because just adding these 5 residues back causes increases in human CIRBP expression, which of course is very similar to what we would see in whales. And again, this cold-inducible RNA binding protein facilitates non-homologous end joining and homologous recombination repair, and it reduces these markers of insufficient DNA fixing, such as, for example, micronuclei and insertion deletion formation. And this is especially common, or especially active at 33 degrees Celsius, which of course is pretty cold. Hence the name cold-inducible RNA binding protein. Then it's also found that whale CIRBP has a higher codon adaptation index. I didn't know what that was. And so it actually means that their codons, again, codons are kind of very degenerate in the sense that multiple codons can create the same protein. But theirs are actually more synonymous, which means that they have higher translational activity from mRNA to amino acid, even if they have different kind of degenerate codons that contribute to an amino acid residue. So you can see the formula for that there, how they calculate that codon adaptation index. So then to kind of wrap up this mechanistic discussion, I just want to again go over what non-homologous end joining and homologous recombination actually are. Again, these are both ways how eukaryotic cells tend to repair our double-stranded genomes. Non-homologous end joining, of course, uses the ligation of 2 separate strands generated by double-strand breaks. So it can tend to be a little bit more erroneous, and yet, of course, CIRBP introduces very, very accurate NHEJ for bowhead whales. And homologous recombination, on the other hand, uses a template of complementary strand to repair double-stranded breaks. So it's a little bit more accurate in general. And again, both of these features are something that CIRBP both tend to correct. And then just to wrap this discussion up, they had a really interesting basic science experiment at the very end of this paper in which they actually overexpressed CIRBP, both the human and the bowhead whale forms, within fruit fly species. So Drosophila melanogaster, of course, an entirely different species. And they found that, as you can see in these slides, you can see that overexpression of CIRBP of both the bowhead whale and the human variants tends to increase survival of cells. Even when you introduce really significant genotoxic stress with 650 gray X-ray radiation, for example. And so in both these aspects, you can see in figure B here, in both these aspects, CIRBP tends to account for accurate repair, genomic stability, and decreased, for example, insertion deletion events, micronuclei, and chromosomal instability, even despite the fact that bowhead whales tend to have an increased propensity for oncogenic mutation as compared to humans. And this teaches us that bowhead whales don't just simply rely on apoptosis, but they also rely on really accurate NHEJ and HR via CIRBP in order to correct oncogenic mutation before it fulminates into oncogenesis itself. So very fascinating article about the comparative physiology that can really teach us a lot about cancer therapeutics and cancer genetics just by examining how different species cope with cancer in general. So I hope you learned something from this video, and I really hope you take the time to dive into the article as well. It's always important to read those. And I hope to see you for the next video. Thank you.